So, dear colleagues, today is day for our Kharkiv chemical seminar, and we have a speaker from France, from Strasbourg. He is originally from Kiev, no, not from Kiev, from Kherson, but then obtained his PhD degree in 2003 from Kiev National University. And then he worked as postdoctoral fellow in the University of Strasbourg and Catholic University of Leuven. Then he joined CNRS in 2006, received CNRS bronze medal in 2010, and was promoted to Director of Research in 2014. In 2015, he obtained ERC Consolidator Grant Bright Sense to work on fluorescent nanoparticles for ultrasensitive detection of cancer markers. In 2021, he received prize of Dr. and Miss Henri Labbe from French Academy of Sciences, and he was elected as a member of Academia Europea. He is a leader of nanochemistry, a bioimaging group in Strasbourg University. His research interests include functional fluorescent materials, molecules, and nanomaterials for biosensing, imaging, and in vitro diagnostics. He is a co-founder of startup Bright Sense Diagnostics, focused on molecular in vitro diagnostics based on fluorescent nanoparticles, and Astronice, developing fluorescent biomaterials for image-guided surgery. He is an author of over 260 peer-review articles and 12 patents. And dear Professor Klymchenko, please, you can start your report.
Thank you very much. Thank you very much, Valentin, for this invitation, and thank you for organizing these seminars. I'm really honored to be here with you, and I'm really admiring the fact that you can continue doing science despite all this situation and difficult times in the history of Ukraine. And so, thank you very much. Yes, I would like to present our work of my team, which is actually it has been already since 2014-15 that I established my team, which is now photoactive materials and bioimaging. Before it was named nanochemistry bioimaging, and our main directions are fluorescent molecular probes. And here we work on molecules that help to visualize biological processes at the level of biomembranes and organelles. We visualize RNA inside the cell and receptors and the cellular and animal levels. Fluorescent nanoparticles, organic nanoparticles assembled from polymers and lipids. In particular, application here are biosensing and bioimaging. And the third direction is rather emerging, is fluorescent biomaterials. These are mainly applied for image-guided surgery and biosensing. Yes, so today I will mainly talk about molecular probes and nanoparticles, and I will present different aspects of our research. So, but first, so you see my slides, they're changing, it's all right? You see my slides are switching well. It's okay. Yes. Okay, excellent. Yeah, so I would like to start first with fluorescence. Probably you know it, but I would like to be sure that we are all talking about the same process. So the fluorescence is a singlet transition. It's a molecule that absorbs light, goes to the excited state, singlet excited state, and then it comes back as a singlet transition in form of fluorescence, where it can go and they go into intersystem crossing in the triplet state, and this is something we are not interested in. Then it will be phosphorescence. So we are interested mainly about fluorescence for multiple reasons. First of all, because this is a highly sensitive technique, we can go down to single molecule detection. Then it is very fast because the process of absorption and emission is super fast. So we go to nanosecond and picosecond time scales. It is a non-invasive technique because actually we are not really putting something in the system like we're not touching the object of study. We just shine light and record the information. And then it is highly specific because it is related to particular molecules that emit light. And however, to do this imaging, you need so-called fluorescent probes. And then what is interesting, you can go from a single molecule to cell, tissue, and even animals. And if we talk about molecular systems or nanosystems that used to visualize, so fluorescence require a contrast agent. And this, there are two types. There are molecular probes and nanoparticles. And molecular systems, there are organic synthetic dyes and fluorescent proteins. Those systems are particularly interesting because they're small size. They're just one or a few nanometers. However, and so they are quite interesting for imaging applications, and you will see some limitations, in particular brightness and photostability. Nanoparticles, in contrast, this is something ultra bright. And then when you do imaging with nanoparticles, you actually can see individual objects, individual particles, which is more difficult with molecules. And this is important particularly for designing of highly sensitive biosensors. But their disadvantage is large size, which makes them more difficult applied in cells in imaging agents. So you will see, therefore, we use mostly molecular probes for imaging application in cells and nanoparticles for sensing. So now we come to the concept of fluorescent probes. What is fluorescent probe? Primary, it's a fluorophore, which was just talking about, and it should be functionalized with some units that will make it specific to, for example, targeting unit to send it to a particular compartment of the cell, and recognition unit, which will help to recognize and detect and analyze. And there is also a transduction unit that converts the recognition into a fluorescence response. And fluorescence response can be different. For example, it can be a fluorogenic response, which means increase in fluorescence. So when a light is detected, light goes up. It can be chromogenic, which is change in the emission color. It can be fluorescence lifetime, which means change in the fluorescence lifetime used in so-called FLIM, fluorescence lifetime imaging, and then on-off switching where the molecule goes up and down. And that is used a lot actually in super resolution imaging. If we talk about concepts of fluorescent molecular probes, there are multiple concepts, and it's very difficult actually to classify molecular probes. But I will try. We could consider two. One is probes which operate by covalent approach. And here, there are mostly fluorescent labels could be considered. For example, fluorescent labels of proteins that operate by copper-catalyzed cycloaddition reaction, like click reaction, or for protein tags. These are molecules that react with specific proteins by enzymatic reaction that would make auto-labeling. Then it can be a non-covalent approach. And here there is a lot of examples. These are mostly, we can call them real probes, because those molecules, they will go at the right place and sense something locally. For example, they can go and co-assemble with lipid membranes. They go in specific organelles of the cells. Or, for example, we could consider probes that do molecular recognition, for example, ligand-receptor interaction, or they could recognize, for example, nucleic acid, specially designed like aptamers, or specially designed fluorescent activating proteins. And I will mention a little bit of these both approaches. Now, when we go back to fluorophores, there are two types. One is classical fluorescent dyes. So here we can consider first, these are the most fundamental classes of dyes, coumarins, body pyridamine, cyanines. Actually, most of examples that you will find in literature of classical fluorescent dyes are those coming are derivatives of those families. And characteristic feature of those, they are very bright and they are not sensitive to the environment. They always emit more or less at the same wavelengths with similar fluorescence quantum yield, independently of the environment, polarity, viscosity, etc. But then you have another peculiar class, and we are particularly interested in those molecules. These are environment-sensitive dyes. And there are different concepts of those environment-sensitive dyes. Generally, these are molecules that detect their molecular environment and they change the properties as a function of this environment, like polarity, viscosity. Here, the first, the most probably famous class are so-called Solvatochromic dyes, which are push-pull molecules sensitive to polarity. There are also so-called dyes undergoing proton transfer. I worked a lot during my PhD, actually with my colleagues, my supervisors, Professor Pivovarenko and Demchenko. We worked a lot, and it continues this activity now worldwide. Dyes undergoing proton transfer. They are sensitive to hydrogen bonding and polarity, for example, by changing their dual emission. Then you have so-called molecular rotors. Those are molecules that undergo excited state rotation, and they change their intensity as a function of viscosity. Then there are flippers. These are a particular class that do a rotation in the ground state, and their emission, excitation, and emission depends on their molecular ordering, particularly in lipid membranes. And then another class is fluorogenic dimers. Those are molecules that, for example, in water can be in a closed state, not fluorescent, and in, for example, organic solvent, they open up and they light up. These are fluorogenic molecules. Today, I will present you examples of solvatochromic dyes, molecular rotors, and fluorogenic dimers. So let's go first to solvatochromic fluorescent dyes. So those are usually a push-pull structure. You have an electron donor, electron acceptor connected with an aromatic structure, polycyclic. And these molecules, for example, like NiRad, undergo in the excited state a charge transfer. So the charge is moving from donor to acceptor. It creates a push-pull, highly dipolar molecule. It interacts with the polar environment, for example, water. And then this strong interaction decreases the energy of this singlet excited state. And the increase in the energy means longer emission wavelength, so that when you increase polarity, their emission shifts towards red. And this can be, for example, shown here, where you have a flask with hexane and NiRad inside. It's blue emitting. And then we start adding tetrahydrofuran, which is polar, and the color switches because polar environment shifts to longer wavelengths emission. And this is essentially because of dipole-dipole interaction, electric fields, and hydrogen bonding. And there are many different environment-sensitive dyes reported, solvatochromic dyes reported to date. I here present a list of those most common classes. They are always characterized by a donor, acceptor, and aromatic connection. And in particular, the most famous ones were this small molecule called prodan and its derivative lordan, and NiRad. So you will hear some examples of NiRad in particular. And at the early times, we actually found another relatively small push-pull molecule, which was actually showed remarkably high sensitivity. This is a sensitivity scale. This is NiRad. This is prodan. And so we actually we developed one of the most sensitive molecule based on this push-pull fluorine. And in fact, this was the Ukrainian student, Kucherak, one of my first PhD students, who did the synthesis of this push-pull dye from bromofluorine, methylation, then nitro group was inserted to reduce to amino group. Amino group was alkylated. Then brom with butyllithium was replaced with the aldehyde function. And then to make a ketone, we use Grignard reagent, and then oxidized. And so we make aldehyde as acceptor and also ketone as acceptor. So with these two. And what is interesting that when we compared this push-pull fluorine versus push-pull naphthalene, Prodan, and this is our new molecule, we observed that the absorption was strongly shifted towards 400 nanometer, which was important to work invisible, and the emission was highly solvatochromic, much more solvatochromic than that of Prodan. And we can also see this on the dependence on polarity, much sharper compared to Prodan. And actually, this molecule beat this commonly used molecule in everything. It was extinction coefficient was higher, dipole moment, transition dipole moment was higher, two-photon absorption was higher. Everything was better with this molecule. And another example that we actually introduced was to go towards near-infrared, a push-pull structure that can operate in near-infrared. And this was actually done in collaboration with Yuri Kovtun in the Institute of Organic Chemistry in Ukraine. He was particularly interested in some deoxyribonucleic derivatives, and we make a push-pull structure, a much longer one. And so here it's actually synthesis. First, we had to synthesize the aldehyde of this dialkylaminobensphoryl aldehyde, and then this shorter version of deoxyribonucleic, and then the Knoevenagel condensation give us this long structure, conjugated push-pull structure. And this molecule beat NIRAD in terms of all properties again. So NIRAD was another reference commonly used. Again, we had a huge shift towards red, and sensitivity to polarity was much higher, and molecule went to near-infrared, so we were already above 700 nanometers in emission. The absorption was shifted to red, extinction coefficient was much higher, two-photon absorption was much higher than red, so everything was better with this molecule. So important to get those structures, you need a high conjugation, high electronic polarizability, and push-pull structure, like it's shown here. So now, what are those molecules can do, those solvatochromic dyes? They are particularly interesting for biomembranes, because biomembranes are polar structures. And biomembranes are very interesting targets for biologists also, and for biophysicists, because membranes, biological membranes, are everywhere in the cell. If you look at the structure of the cell, you can recognize actually that almost any organelle is surrounded by a biological membrane. It's essentially a lipid bilayer with proteins. So anything, mitochondria, lysosome, cell plasma membrane, nucleus, all surrounded by membranes. And those membranes, they play multiple functions. They're natural barrier separating exterior from interior, or interior of organelle from cytosol. They transport nutrients, they do undergo signal transduction, respiration, photosynthesis, electrical activity, lots of things. Another interesting thing is that a lipid membrane is very complex, and it has a not at all simple organization. Even lipids are specifically organized into microdomains of a certain type of lipids, surrounding particular proteins, making kind of factories of proteins to do particular function. And therefore, tools to study those membranes, structure, organization in the cell is very important. And so here comes the solvatochromic probes, which are particularly suitable for those membranes. What happened when we take a NIRAD dye, which is highly solvatochromic, and add it to lipid membranes? So when it is in water, water is highly polar, so the dye is emissive in rather red region, and it is not very bright. It's quite quenched by water. When it goes to membrane, which is essentially composed of lipid fatty acid chains, highly apolar environment, it will shift to the blue because the environment is highly apolar, and it will increase its intensity. And this is essential signature for us for imaging of membranes and for probing their properties. And throughout these years in our lab and also worldwide, there were groups working on different types of membrane probes based on those push-pull solvatochromic dyes. And so the primary is Laurdan and NIRAD, but then there are many other examples. And so my team developed a number of those structures where we have a solvatochromic dye, and then a characteristic structure is a polar group and a chain, which makes them particularly affine to lipid membranes. And this is an example. This is again the same student like Skucharak, who actually designed a derivative of N12S, NiRed, conjugated with their anchor group. And you can see that when this molecule is added to the cells, we see a very beautiful staining of plasma membrane. While in case of non-functionalized NiRed, it goes inside the cell and binds everywhere. For example, these are lipid droplets inside the cell. So this functionalization is key to achieve specific staining of plasma membrane. And then these molecules are chameleons, so they change their emission color as a function of lipid organization. For example, if you take in lipid membranes, there are so-called liquid ordered phases and liquid disordered phases. Ordered phases are composed of saturated lipids and cholesterol, like sphingomyelin cholesterol. Disordered phases are unsaturated lipids with a little amount of cholesterol. So when we compare spectra of this probe, ordered and disordered phase, they are different in polarity. This highly ordered structure do not contain a lot of water, so it is shifted to blue, compared to the structure which is loose, with many pores, water enters, shifts to red. And now when we have these shifts, we can actually transform them into color images by doing imaging with using microscope and splitting the spectrum into two channels, the blue part and red part. Then we divide one channel by another, and if the emission shifts, the color will change. Example is, you can see here, in case of giant vesicles. So giant vesicles are like artificial cells. You have a lipid membrane and in form of a vesicle of a size similar to the cell. It's like 30, 50 micrometers formed by electroformation method. Then when you add this dye to those vesicles, we can see that vesicles of different lipid organization are colored in different color because the ratio here that we generated in our microscope are different. If you mix two different phases, you see those domains. So these domains were hypothesized to be present in the cell as so-called lipid rafts. However, when we use our probe in living cells and we did this ratio image, we didn't see any rafts. In fact, all cell membrane was quite homogeneous. It was a big disappointment for us, but we could, for example, see that if you remove cholesterol, you change order and you can see change in the lipid order. But rafts were not there. And in fact, this remained to be a hypothesis, and it's very difficult to access them because they are very small and dynamic, those lipid domains. And so we thought maybe we can develop probes for high resolution imaging and go to nanometric scale. And to do that, people develop so-called single molecular localization microscopy or nanoscopy. But for this purpose, you need dyes which are undergoing on-off transition. So in this case, what happened, if you do single molecule localization with those dyes, most of the image, you see only few molecules. And you shine light and you continue recording images of a few molecules one by one. And then you can localize them in space with nanometric precision. And then you can reconstruct the image and form so-called super resolution image. To show this is a little bit a French version of how to show super resolution. You see the on-off switching in event, you localize each molecule, and then you reconstruct the high resolution image. And this is an example of a low resolution image of actin filaments. And this is already high resolution using this method. And in fact, we were particularly interested to make on-off switching using so-called paint approach, where the molecule on-off switch by binding. So in water, it is not emissive. And when it binds to membrane, it becomes emissive. As I said before, it is a property of NiRed dye. But NiRed goes inside the cell. So what to do? So what we did, and that was done by Dmytro, another Ukrainian student, a PhD student in my team. What he did, he replaced this anchor group with much shorter chain. So this shorter chain ensures that the binding to the membrane is reversible. So the affinity of this guy is low, so it should bind and unbind, unlike the original N12S, which binds irreversibly. So when this was done in collaboration with Keng Su in Berkeley, when we added this dye to a membrane on the surface, so-called supported lipid membrane, and we run video imaging, you can see those little dots. Those little dots are individual molecules that bind and light up when they bind to their membrane. So this is perfect for super resolution imaging. And so we could obtain now the cell surface. So this is a cell, this is a membrane, little protrusions, and even a little endosomes we could localize with nanoscopic resolution. And as this molecule is chameleon, we could actually extract spectral information. This was used using prism, and each single molecule give a spectrum that can be analyzed and transformed into color. And so color here reflect lipid organization. So we could achieve and see those nanostructured domains of different lipid organization on the cell membrane, thanks to this combination of solvatochromism and reversible binding. But then we wanted to go further inside the cell, because the lipid membranes are different on the surface and inside. For example, 60% of all cholesterol is on the plasma membrane. So that inside there is only little amount. So lipid organization in each organelle should be different. And so what we did, we took our NIRAD as a starting point, carboxylic derivative, and we start designing molecules that target different compartments of the cell. So this one I already showed, it goes to the plasma membrane. But then we functionalize this with a morpholina group, which can be protonated, and this by so-called proton sponge phenomenon will accumulate in lysosomes, which are very acidic compartments. Then we attach highly hydrophobic and bulky group to go to lipid droplets, a cationic group to go into mitochondria, then thiol-reactive groups, which is known to target endoplasmic reticulum, and lipid functionalized group that can go to Golgi. So we exploit natural biological processes that hijack the molecules. And so like this, we can localize them in a specific compartment. So the synthesis of NIRAD, we are using already established protocols. So the first primary, we had to get this phenol having carboxylate. Then we need to make a nitroside derivative. And then with alpha naphthol, there is a condensation reaction. It's a multi-step process giving a NIRAD. So then NIRAD carboxylate is coupled to different amines, either this anchor or different organelle targeting groups to get these molecules. And then we go from the chemistry lab to cell culture, and we study cells using fluorescence imaging. So this is confocal microscopy, and the first example is membrane probe, and we compare with a reference in green, and our new probe is in red. And so you can see the membrane probe targets perfectly plasma membrane because it localizes with membrane targeting reference probe. Then the ER targeting NIRAD, the formula shown here, they target ER, which can confirm by the green emission of ER tracker. The same for mitochondria colocalization is ER tracker. For LISA tracker, it works too. For Golgi, there is colocalization too. And for a lipid droplet, probe goes to lipid droplets. So what we show that by using different targeting group, we can send NIRAD to different compartments at will. But in fact, this is not should not be taken for granted. You cannot just take organic molecule and send it to any compartment. It is particular property of NIRAD that helped us to realize this. So now, as we are able to send this probe to different compartments, now we wanted to know the polarity, local polarity lipid organization of those structures. So we again did ratiometric imaging, and this color reflects local polarity. So red is high polarity and blue is low polarity. The least polar was lipid droplets, which is normal. It's a core is oil. Then it was plasma membrane. And then intracellular organelles were much more polar because they have less cholesterol. However, when we applied hydrogen peroxide, this is an agent that produces oxidative stress, in particular lipid peroxidation, which introduces hydroperoxide groups, highly polar groups. It provoked increase in polarity of different organelles. Not all, but some organelles like lipid droplets, Golgi, and mitochondria. So some organelles are more sensitive to oxidative stress than their others. So now I would like to switch to molecular rotors. So those molecules, as I mentioned before, they undergo rotation in the excited state. And because of this rotation, they are quenched. And so in low viscosity solvents, just usual organic solvents, they are almost non-emissive. When we increase viscosity, their fluorescence emission and lifetime increases. So they are kind of fluorogenic. They light up at higher viscosity. At this point, we were thinking to develop different types of rotor molecule and apply them in biological application. And one of them was deoxyborine, again in collaboration with a team of Kovtun in Kiev, and actually another graduate student, Karpenko, who worked here in collaboration also with Dominique Bonnet group. So what we did, we prepared deoxyborine, which is push-pull, but it is shorter than the one I showed before, and it had a very high sensitivity to viscosity, and this sensitivity to viscosity was correlated with the viscosity perfectly, linear correlation. It was highly photostable also compared to NARED. And then we say, how about we can develop a turn-on probe based on this? Because the environment in water is non-viscous and in lipid membrane is viscous. So we thought we could apply it as a turn-on probe for membrane receptors. So what we actually should do is to couple our molecular rotor to a ligand, and this ligand receptor binding would drive the fluorophore into membrane, which is highly viscous. It will light up. So this was done in collaboration with Yulia and Dominique Bonnet, Yulia Karpenko and Dominique Bonnet. And they had a model for those receptors and a ligand. So they had a magic ligand, which is carbetocin, which recognized GPCR, G-put coupled receptor of oxytocin. So we prepared first this deoxyborine carboxylate and coupled it to this cyclic peptide, which targets receptor. And you can actually see that in the presence of the target receptor, you have high fluorescence. And here, these are in the presence of competitor that removes our probe from the cell membrane, which means it is specific to receptor. Yeah, and another application we thought for fluorogenic viscosity sensitive probe is to detect specific lipids. I was talking about before about lipid organization, but how about to target specific lipid and detect it? And so what we thought it would be interesting to detect phosphatidylserine, because in a normal cell, healthy cell, there is no phosphatidylserine in the surface. However, during apoptosis, scramblase enzymes, they will bring phosphatidylserine from inner leaflet to the outer leaflet. So, and then if you can detect it, we could detect apoptosis, so-called programmed cell death. It's important in cancer research, for example. And for that purpose, we thought to take a molecular rotor molecule, couple it to a receptor of phosphatidylserine through a spacer, which is PEG spacer. And so there, we thought that if a receptor recognizes phosphatidylserine, the rotor will be inserted in viscous membrane, and this would light up. So this probe was sensitive to viscosity, which was good news. And then, so how we did that? So this is an example where you need multi-step synthesis to arrive to this final probe. In particular, first we had to synthesize body pi molecule. That these are standard procedures from aldehyde and pyrrole. But also then we need to attach a linker with the azide to do further click reaction. And then we also build our receptor unit. So this is a multiple alkylation reactions. And then after we attach the acetylene unit. And then we combine these two, fluorophore and receptor together. And for this purpose, we use click chemistry. It's particularly efficient to use click chemistry when you want to combine two big molecules with good yields. And then we add zinc to form the final receptor zinc complex. So then we went again to their biolab and we did experiments on cells. So we provoke apoptosis of cells using staurosporine and then image. We also had to use Nexin-5, which is a reference molecule to see apoptosis. And so what we were happy to see is that our zinc probe, which is in green, colocalized with the commercial probe, which is in red. So this is an apoptotic cell, and it is labeled with our probe. And other cells, which are not apoptotic, they are not labeled. Another interesting feature that our probe also saw late apoptosis because it internalized in those apoptotic cells, which was not the case of annexin. So we actually developed molecular tool to see apoptosis and early and late using molecular rotor. So the final type of probe I wanted to talk about are fluorogenic dimers. This comes from a phenomena of so-called aggregation cost quenching, where you take fluorescein, if you put it in solution at high concentration, it loses fluorescence compared to molecular form. And this aggregation is come from the Kasha theory, exciton theory, which says that when molecules assemble face-to-face like this, organization is so-called Ash aggregate, which is forbidden, which is a forbidden transition for lower state. It can go to the higher state, but then it goes to lower, and their emission is forbidden. So they are quenched. The G aggregates are allowed. And we thought to use this Ash aggregation to develop probes. For example, intramolecular system that could be closed in water and turn on when it binds to target. And one of examples is to use it for super resolution imaging of membranes. So what we wanted to have is to have a dimer of the dyes, which is self-quenched in water and light up when it binds to membrane, similar to what we did with NARED, but in this case, it's two highly emissive cyanine dyes which we used. So you can see two cyanines connected with a lysine unit. So these are anchor groups, but low affinity, so that we can have reversibility. So what is nice is this dimer had a characteristic short wavelength band, which correspond to Ash aggregate. So in water, it is self-quenched. When you put it in ethanol, absorption looks like a monomer because it opens up. And in fluorescence, it was poorly emissive in water. This is what we wanted because it is in this form. And when it is in ethanol, it opens up and increases fluorescence. So the system is fluorogenic. And then this is again in collaboration with Kiksu and Brackley. We added to the cells. This is a reference NARED, and this is our butterfly probe. So what we can see is that this molecule blinks. This is what we need. So it on-off switches when it binds, but it is also much brighter than NARED because it has two super bright fluorophores that lights up together. And because of these properties, it allows us to do much higher resolution imaging of the plasma membrane compared to NARED. So threefold higher improvement. And it is also, because it's brighter, higher resolution and faster. This video shows actually that we can image at high resolution the cell surface. So these are individual extensions of the membranes, which are highly dynamic of the cell surface. Another application of this dimer, so that was just binding, not specific binding to cell surface, but we sought to couple it with a receptor recognition, as we did for molecular rotor. And for this, again, this was in collaboration with Dominique Bonnet and Yulia Karpenko. We designed the dimer, which is in this case composed of two squarane dyes. Squarane dyes with long linkers connected again by lysine. And from this side, we attach carbitocin, again, to target a G protein couple receptor. And our hypothesis was rather simple. In water, they are self-quenched. And then when ligands bind to receptor, it will open up on the membrane and it will shine on the membrane. And this is what we observed. So when we added this probe to cells, we could observe a strong emission for the cells which expresses the receptor. I don't have time to show all controls, but if there is no receptor, you don't see fluorescence. But what is important, when you take a classical dye, which has a ligand and fluorophore, which is always fluorescent, you can see a lot of background because the probe, when it is not bound, it is fluorescent. And when it is bound, it is fluorescent. So you see fluorescence everywhere. This is an advantage of using this on-off probe to have no background. But this is also advantage to do the imaging in mice, because when you inject in mice, you cannot wash out a non-reacted probe. It will fluoresce everywhere. So to have a turn-on system, it would be advantageous to target a receptor. So, but to use mice experiments, we needed to design probe which has much longer conjugation. So this was a cyanine 5.5 dimer, which operates in near-infrared region. And near-infrared is good for mice experiments because we can penetrate deeper with light, and so we can see tissues inside mice. And in this case, we use lactating mice, which actually gave birth, and so she was lactating. And then it overexpresses naturally oxytocin receptor at mammary glands. So we injected the probe, and we could see mammary glands shining, which means that we targeted receptor there. When we added free carbetocin, we didn't see any fluorescence, which means it replaced locally there from the receptor. And control mice, which was not lactating, did not show fluorescence from mammary glands. So it was receptor-specific. In fact, it was the first example where we could, where the receptor naturally expressed could be targeted and imaged. So, so far, I showed you non-covalent approaches to label membranes, membrane proteins or lipid membranes. And all these non-covalent approach, you see all these examples, has one problem. When you go to a natural medium, for example, serum, or you do fixation permeabilization, non-covalent probes will desorb. So, for instance, we develop membrane probe, which is binding, but it binds reversibly. It will not work in natural medium containing serum. So therefore, we recently introduced a concept so-called lipid-directed covalent labeling of plasma membrane. And it is based on a dual principle. One, you have a ligand to bind reversibly, as I showed for super resolution imaging. So this is a little anchor group. And the second, to have a reactive group. In this case, it's NHS ester, so activated carboxylic acid. And the idea was that it binds, concentrates on the plasma membrane, and then because of high local concentration, could react with amino group of proteins and provoke covalent modification of proteins. And so we designed this molecule and added to the cells. So this is based on cyanine. And we actually were happy to see that plasma membrane was staying very well. And then to understand what we got, we start trying control. So control where COH group was not activated did not label anything because it cannot react with amino groups. So it cannot label. Then we tried another activation using pentafluorophenol. It is much less reactive. It could not label the membrane. It internalized. Then we tried actually a version without the anchor group, without chain that binds to membrane, and labeling didn't happen. We had mainly internalization. And then we took a commercial NHS ester. So this is highly polar label for amino group. It didn't work either because or it worked, let's say, very weakly because we did not have membrane targeting moiety. So we need these both functions together. And what is interesting is now we are able to label covalently, permanently, the plasma membrane. And then when we put it in serum, we can observe for a long time fluorescence on the membrane without detachment. However, if we use, for instance, this membrane probe that binds non-covalently, it detaches because serum takes it back because it desorbs. So we can do long-term imaging. And we can do more than that. We can, for example, label some cells in green color with cyanine 3, label other cells with red color, and then co-incubate them together. And you can see that after 5 hours, individual color observed, but even after 29 hours, green and red cells are separately seen. So we can track individually the cells in different colors. And this is, for example, a video that is recorded for instance, this is within 1 hour scale. We can follow movement of the cell. We can see the dynamics, and we can see interaction of red cell and green cell over time. This would not be possible with other membrane probes, which are non-covalent. But even we can do further, and we can start mixing colors. For example, we have a green and red, and we can, like a printer mixes colors to make multiple colors, we mix green and red in different proportions, 100%, 50-50, or 75%, or red or green. And you could see that you can generate almost a rainbow colors through this covalent labeling. And then when you mix cells and incubate for for multiple hours, you can identify those individual barcodes. For example, green, you can see lime, you can see orange, yellow, red. They coexist together and they don't exchange because of covalent linking. Okay, so I finished my part on molecular probes. So I was presenting different environment-sensitive systems, solvatochromic dyes for membranes and organelles, molecular rotors for receptors and lipids, and then fluorogenic dimers, mainly for super resolution imaging and for their receptors. And so importantly, that many of those probes were commercialized, and actually many of them are used worldwide in more than 500 labs. Okay, so now I switch the gears. This will be much shorter part about fluorescent nanoparticles. So here I want to just present a few examples. What is important? What is motivation to work with nanoparticles? These guys are far brighter than organic dyes. You cannot you can improve fluorescence brightness of organic dye 10%, 20%, maybe twice, but you cannot improve it 1,000 times, or 100 times. Fluorescent nanoparticle can do that. Quantum dots are actually one of the best, but then you have other examples that appeared in literature, like dye-doped silicon nanoparticles, some gold cluster carbon dots, aggregation-induced emission particle, conjugated polymer nanoparticles. We were particularly interested in two classes: lipid-based particles and polymer particles, dye-doped polymer particles. In case of lipids, we were interested in the lipid core as a nanoreservoir to make supramolecular probes. And in case of polymer particles, as bright objects for biosensing and diagnostics. So I will show you two examples of this each. So for that, let's go back to concept of fluorescent probe. As mentioned before, you need a recognition unit and fluorophore. And our target in this case is a dopamine. Dopamine is a neurotransmitter. It's super important in neuron-neuron communication, in behavior, etc. And it is very difficult to build a fluorescent probe for dopamine. In literature, there exists this kind of example, which can form imine bond through aldehyde with fluorophore, and it recognizes catechol unit through boronic acid. And then it forms this complicated cycle, which is a kind of receptor that change fluorescent properties. However, this molecule works not so well in water because imine bonds are not stable in water, and so affinity is very poor. So they cannot be used in cells to detect in the dopamine. So we thought to rethink the concept, and how about to use those lipid droplets, which can be prepared by simple mixing of oil, surfactant, and dye in water, and you can generate a nanoreactor where the oil core could be used to do biosensing. And the first actually observation that we did, which motivated us, was we mixed this push-pull aldehyde molecule with an amine, and we observed imine formation. Imine formation was observed as change in the color of the solution, emission color, because ketone or aldehyde is a strong electron acceptor and imine is a weaker electron acceptor. It changed emission properties. So this imine formation can only happen inside nanoreactor. It cannot happen in water. And we said, okay, maybe we can design a supramolecular sensor where you have a fluorophore aldehyde, which contains also a recognition unit. And then when we add neurotransmitter, recognition unit will capture neurotransmitter, bring it to the nanoreactor, and then amine will react with aldehyde. And so this was our aldehydes. So this was the original aldehyde, and this was designed, special designed aldehyde. Recognition unit, again, boronic acid, and this is our target, dopamine. So this was done by Bogdan, so another Ukrainian student. This is in collaboration with Jean-Marie Lelab. And so first we tried pyrene. Pyrene actually does not react with amine very well. It takes 24 hours that imine is formed. We took very hydrophobic amine, which goes directly to droplet, and imine is formed in 24 hours. So for sensor, it's a disaster. We wanted fast imine formation. So for that, we redesigned an aldehyde, which is in orthoposition to pyridine unit, a push-pull structure. And then this molecule reacts much faster with amine, form imine within 20 minutes. So we use this molecule in the next steps. So if you just have an aldehyde inside their oil droplets, it will not recognize any of those neurotransmitters because they are water soluble. They cannot go into oil phase, so you cannot react. So no response. However, when we added a recognition unit that can recognize dopamine catechol unit, we start observing response to dopamine. And the response was only to dopamine because only dopamine had these two OH groups. So we observe a nice fluorescent response, a shift to the blue. Imine is formed because of molecular recognition. And when we tried others, response was almost not there. The exception was noradrenaline. It had also two OH groups. And sensitivity was quite good. We did this actually calibration. It was in a micromolar range. And this allowed us, so the shift to the blue was increase in the concentration of dopamine. And this allowed us to do imaging of dopamine in their microfluidic channel. So we added dopamine from here, and then it creates a gradient. Here, no dopamine, here there is. And so it creates a gradient. This is high concentration of dopamine, this is low. It's a ratio imaging. You can see change from red to blue. And then this is a gradient, so we can follow dopamine concentration in the gradient. So this was an example of using oil droplet. However, the other example is to use a polymer particle as an ultra bright object for diagnostics. And yeah, so this will be my last example of material and application. And in diagnostics, what we want to do, we want to detect biomarkers of a disease. And the typical biomarkers are, for example, nucleic acids. We all know that in the period of COVID-19 that PCR was very important because we detected a marker which was RNA, a viral RNA. And what is really attractive is to do so-called point-of-care diagnostics, where diagnostic could be detected, for example, by generalist or by or even at home, you can detect a virus or a cancer marker. And so we were thinking to detect nucleic acids using our bright nanoparticles. And why you need to use nanoparticles? In fact, if you want to detect nucleic acids in biological fluids, for example, in this nasopharyngeal swab or in the blood, you need to detect something below 1 picomolar, even femtomolar. It's very low concentrations. The modern fluorescent techniques using commercial setups, they allow you to detect only nanomolar. So there is a huge gap of 1,000 or million in sensitivity. That is why people use PCR. They amplify the number of targets. And after you add fluorescent molecules, when you amplify million of times, you can detect. And so we thought maybe using our bright nanoparticle, we can replace non-enzymatic method with our method, which will be simple and direct. And for that, we need to go through several challenges to develop nanoparticle. First, to make polymeric nanoparticle, we need to assemble it from polymers. And to make them small, so this is just briefly, we used charged hydrophobic polymer with a single charge per chain. And then by nanoprecipitation, we could form very small particles in the range between 5 and 14 nanometer. The second challenge that we had to go through is the fact that dyes, when you encapsulate them, they don't fluoresce, they aggregate. As I mentioned before, they just stack and then switch off because at high concentration, they will like each other and form ash aggregates. So we need to prevent so-called aggregation suspension. And then to make sensors, we need to make them, for example, undergo energy transfer. But this is not easy because particle is too big. And the FRET, for example, the first resonance energy transfer works at 5 nanometer. Our particle is 40 nanometers. So how to address? So I will briefly mention these two, what we did. How to prevent self-quenching of dyes in particles. Nature resolved this problem in chlorophyll. They use light harvesting complexes where porphyrins are separated by proteins. And this allows actually the life on the planet converting light into the reaction. They deliver energy into their photosynthetic center. What chemists do? Chemists, for example, synthesize dendrimers and they separate fluorophores in space. Or, for instance, they design so-called aggregation-induced emission dyes, like one example is here. Or they put bulky groups on a fluorophore. Bulky groups separate fluorophores. And our approach was different. We like rhodamine dye, super bright dye, but it's cationic. Why it aggregates? Why two cations get together? Because you have contraion, perchlorate. So small perchloride brings them together and they stack and then they are self-quenched. So we said, why not to replace perchlorate with a very bulky contraion tetraphenyl borate derivative? And this turned out to be a very efficient approach because it could separate the fluorophores in space, prevent cell quenching, and help to encapsulate. And this is example how particle is prepared. So what we do, we take a rhodamine dye, we pair it with a bulky contraion. It's fluorinated, that's very important. This ion pair can be purified by column chromatography, actually. It behaves like a molecule. We mix with polymer in acetonitrile, we nanoprecipitate and get particles. When we increase loading, the brightness increases. If you use perchlorate instead of this guy, instead of tetraphenyl borate, the fluorescent drops because of self-quench. So we can increase brightness by increasing loading. And then when we compare this with quantum dots, so those semiconductor quantum dots are super bright. When we look under microscope on the glass slide, our particles look much brighter than quantum dots. And in fact, our first example was 6-fold brighter, and our more recent example are 100-fold brighter than quantum dots because of thousands of dyes per particle encapsulated. And so now to go to FRET, can we convert these particles into sensors? So FRET system is typically 2-5 nanometer because FRET is limited by distance of about 5 nanometer. But particle is at least 20-30 nanometer. What to do with that? And in fact, by serendipity, we observe that our particles, when they are on the glass slide, we do video, they blink. They blink like quantum dots. And in fact, more dyes you put, more they blink. And what happened? Actually, exactly like in a light harvesting system, at high density of dyes communicate, the energy migrates. And then when you have a dark state, it quenches all system. So in the chlorophyll, it is happening in this way. So the fluorophore communicates and then goes to reaction center at long distances. And in fact, thanks to this approach, we developed so-called light harvesting nanoantennas based on our particles. They could amplify 1,000 times the emission of acceptor because of this light harvesting principle. And then we converted that into a sensor, into a FRET-based sensor. And this is example of nucleic acid sensor based on this nanoparticle. For that purpose, we needed to functionalize nanoparticle with DNA. Then we added a coding DNA that will recognize our target and hybridized it with a smaller DNA, which has acceptor. So this system undergoes energy transfer, so it emits in red. When we add target, target displaces the acceptor and it turns green. So the color switch will tell us target is there or not. So this was done actually by Nina. So another Ukrainian student actually worked, a PhD student in the lab. So what she did, she designed a polymer containing azide group and carboxylate, a charge. So when you nanoprecipitate, you obtain small particle where carboxylate group brings azide on the surface. So then we could use DNA by click reaction, in this case, strain-promoted click reaction. DNA could be attached by click to the surface, and then we make a FRET pair by hybridizing with a small acceptor. And so the sensor works this way. First, FRET system, you have a FRET acceptor emits. Then you add target, target displaces the acceptor, and FRET disappears. And it is actually sensitive to even to mutations. And when you put this on the surface and look under microscope, you can see acceptor, which is red. This is a FRET system. The donor is green. You see both. So it is in orange color in overlay of two images. However, when you add target, acceptor is removed and it changes color. And we could, using a nanoantenna, we could even image their individual process of hybridization, dehybridization. When it hybridizes, it becomes red, and then dehybridized, it becomes green. So this technique was very sensitive. We went down to 0.25 picomolar, and it worked in serum, which was a starting point for development point of care. And so the last point is that this energy transfer could be even done between particles. You have a particle donor and particle acceptor connected by DNA. And we wanted to see if we can transfer energy to sufficiently long distance. And surprise was, if you use two particles as donor and acceptor, we are breaking the law of Förster, because we can do energy transfer at much longer distances, at 20 nanometer, for example. And this allows us to make sensors for example, where DNA brings two particles together, and then energy transfer appears when we add target. And in this case, we obtain very low sensitivity, very high sensitivity at 0.36 picomolar, and it was much faster than the previous assays. Okay, so I came to conclusion. So we work on different systems, and particularly here on polymeric systems, where we could design very bright dyes. They actually present light harvesting properties. They can undergo energy transfer, which can be used for sensing and sensing through long distances. This part we applied for COVID detection, which I didn't have time to talk about. But in fact, this technology for diagnostics gave a creation of a startup, which called Bright Sense Diagnostics, which develops now the assays. And now I would like to thank actually my team, which worked since already quite a long time. And there are a lot of students, PhD students and postdocs, which contributed to this work. And you notice there was a big crowd of Ukrainian students, which probably contributed to almost 50% of examples of research I was talking today. There are some collaborators, local collaborators, also external collaborators, some of them from Ukraine as well. And I would like to thank the ERC grant, which really generously supported the research, and other University of Strasbourg, Einer, etc., and the CNRS. And thank you very much for your attention.
Thank you very much for your very nice presentation and for a lot of Ukrainian students and PhD students in your group. So, dear colleagues, do you have questions to our lecturer? Please.
A lot of information.
Yeah.
It may be very clear, so there are no questions.
Igor Komarov.
Ah, yes.
Just a second. Can you hear me?
Yes.
Hi. Andriy, I'm very happy
hello.
to see you and listen to your fantastic lecture. The last examples are really fantastic indeed. Congratulations to your successes and many successes ahead. My question is, you said that you group commercialized some materials. Could you add a little bit more details? What is that, and what is the stages of commercialization? Perhaps give us some advice how it could be done and should not be done. Yeah. Thank you.
Yeah, in fact, I can tell you that I am really I really did a bad job in commercialization initially, because we developed a number of membrane probes which would be interesting for commercialization, but I kept on sending them for free worldwide. So when I wrote 500 labs, it's actually manually sent to different labs until the moment, and which was really crazy in 2010. However, at one point, we developed probes which are called MemBright, and those are even more simple probes. They were just based on cyanines, and they were very easy to use. You just add to the cell and you see membrane. And then when we presented this work, people said, "And come on, this will be a huge market. You have to commercialize." And we immediately had many demands, etc. So we approached, we were actually approached by the companies, by multiple companies when we published, and they asked us if we are interested in commercialization. So we selected one, which is cytoskeleton, which works in US, but they were relatively simple in terms of rules for commercialization. So what we agreed that we send them the compound and they make packaging. We just need to do quality control. And so we produce the probe, we send them the solution, and they do all the rest. We just need to provide the quality. And then we did together the advertisement, and then it works. And in fact, now, since we started, then we realized it is relatively simple. So now we already have 10, and I think there will be many more commercialized. And those companies, they're quite easygoing. They are also Merck can do that quite simple way. So this can be done as soon as there is a market, there is interest of at least, how to say, you have at least 5 demands after you have a paper, they want you to send a probe. There is an interest to commercialize. But also another interesting thing about commercialization, it increases impact of your work because visibility goes up. For instance, people in the US prefer to buy than to take for free. And so they start buying, and this increases visibility of research article also.
Of course. Yeah. Great. Thank you very much.
Yeah, thank you, Igor.
Thank you
Happy
very
to
much.
see you personally. Yeah.
Yeah.
Maybe in the future. Okay. Thanks.
Yes, I hope very soon.
Alexander Kirichenko, please.
Okay, Andriy, thank you very much for your interesting presentations. I'm also impressed to see so many amazing fluorescence probe. My question about this part of your presentations about Covalent labeling of biomacromolecules, bioimaging. So to be specific, I'm interested in your opinion about fluorescence protein as competitor or replacement for synthetic organic dyes. This question is because here in Kharkiv University, we also work for years, synthesize and study new fluorescence probe, proton transfer probe. But last year, we see from collaborations with colleagues abroad that at least biophysics start using fluorescence protein instead of typical Alexa dye. So what is your opinion, perspective?
Yeah, it's a really important point because if I was showing at the very beginning of the lecture, we have those molecular systems and we have proteins. And it's true. In fact, when I started my PhD in 2003, practically everything which people use, with biologists used with fluorescence were organic dyes. And then Roger Chen introduced application of those guys in biology. Once these proteins were invented far ago, but the application in biology for imaging was proposed by Roger Chen, who got Nobel Prize together with other colleagues. This totally changed the world. So it was really very frustrating for me as a chemist to see how chemistry became less important because biologists could do everything by themselves. They don't need chemists. However, it was temporal. It was a period of about 10 years, and everything they tried to do with proteins. And then they realized the problem. The fluorescent proteins are very big compared to dyes. So this means if you label with fluorescent proteins, you increase the size largely of your target, and so you cannot track correctly in some cases. Another problem: fluorescent proteins, they are not so photostable. They are not so bright. And so when we do super resolution imaging, imaging with fluorescent proteins is very difficult. So if you want to do advanced fluorescence, we need very bright and photostable organic dyes. So another example, all examples I was talking about was about sensing environment to make turn-on system, color changing system. You cannot do that with fluorescent proteins because fluorescent protein, the fluorophore is hidden inside the barrel. So it does not sense anything outside, while those other solvatochromic dyes, rotors, dimers, etc., they are highly sensitive. So therefore, when those initial wave of fluorescent proteins now goes down, and there is a, let's say, revenge of chemistry, and so chemicals, example of chemical probes now multiplying tremendously, and I would say there will be many more examples of organic sensors, organic dyes compared to fluorescent proteins. So I would say chemistry has its place, and organic chemistry has its place in the development of those. And I'm quite enthusiastic to continue in this direction. And I invite colleagues to contribute and work on that. This is very, very interesting direction.
Okay, thank you, Raj. So we now also see that we in safe positions because chemistry is still on the market. Thank you.
Yes, it's also on the market and widely used. Take one example, neurons. In primary neurons, you want to image neurons and how they work. for example, those ones are very difficult to transfect. They are not happy if you start transfecting them. So you just add organic dye and you do imaging. It's much more simple. Immune cells, it's even worse. If you study immune cells, if you touch them with anything, you do transfection, they get excited and they change, they differentiate. So primary cells cannot be transfected, very difficult to transfect actually. And so the organic chemistry has huge impact. But there is one more thing. Actually, there is one new direction with a marriage of molecular biology and chemistry, which is so-called tag approach, which I mentioned here. It is snap tag or halotag. So this is a combination of two. So you can have a protein which can very specifically react with a synthetic dye. And so then you can develop fluorescent dyes, organic ones, and combine them for a very specific targeting of a given protein, which in this case solve all problems of existing fluorescent proteins. So again, we can even have very selective labeling using molecular biology together with organic chemistry.
Okay, thank you.
Oh, thank you very much. We have a message to you from Vitaly Klymchenko. Dear Andrei, compliments to your lecture from Roman and Vitaly.
Thank you very much. I could not talk about our collaboration because that was a different subject. It is on delivery that Roman really worked a lot in my team and this collaboration, but that was on gene delivery. So I already talked about so many things. I would be happy to talk one day about it, but I guess you know the works of this collaborative work already presented by probably Roman. Yeah. Thank you.
So, dear colleagues, other questions to speaker? You have.
Yes.
Yeah, please.
I'm very pleased to see such impressive presentations. So thank you for the lecture. And I have the question about fluorescent nanoparticles. As I understand, they are rather big. And my question is, what objects are suitable for them? So maybe those are not permittable, and maybe you have some special techniques to make
yes.
permittability. Yes.
Yeah. So in fact, because of lack of time, I could not really talk about cellular applications of those particles. And however, the size of particles can be tuned, so we can make them relatively small. For instance, those bonds, we can go down to 5 nanometer. But even 5 nanometer is much bigger than organic dye, which is just 1 nanometer. So 1 nanometer dyes penetrate cell membrane directly and can be used inside the cell. 5 nanometer particles cannot penetrate directly the cell. And it's true that I can tell you that our project on DNA, that was this ERC grant on DNA sensing, was originally designed to detect nucleic acids inside the cell. And then we turned it to diagnostics. Why? Because we could not bring them into the cytosol to detect nucleic acid inside the cell. So your question actually is a big challenge to bring particles inside the cell. They need to cross plasma membrane. So what happens usually when we use this particle in cells, they bind to the cell surface, and then the cell engulfs them and eat them. And they accumulate in so-called endosomes and lysosomes. So they will never go into their cytosol, so into their interior of the cell. They will be always blocked in this stomach of the cell, which will digest them. And so the methods to deliver them exist, and we actually presented 2 works. One was where we did microinjection. So you can use a kind of needle that can go into the cell and inject, but okay, this you can inject anything using it. With particles, it works. But another method, which is, let's say, can be more used for many cells, is electroporation. Electroporation, you're applying alternating electric fields very rapidly, and then this will create pores, transient pores in the cell, and these pores allow particles to enter inside the cell. And so we did these experiments, and indeed we can deliver inside. However, of course, electroporation can be a bit toxic for cells because you create pores. But electroporation is used now in biology quite a lot. So this could be one of probably the best methods so far to bring particles into the cytosol. I would select this one if I further want to apply them in cells.
Okay, thank you very much.
Yeah, you're welcome.
Thank you. So I don't see any hands. Oh, in this case, okay.
There is one.
Anatoly. Yes, Anatoly.
Yeah. Thank you very much, Andriy, for your very exciting lecture. A lot of information, and really, even to each slide, there are a lot of questions. But I have only one, which concerned about bioorthogonal reaction and covalent labeling. As I understand, usually people at first needs to modify this protein with one of these bioorthogonal reagents. And what is the difference really if we use some modify amino acids, or what is the inverted there, these amino acids, or if in this case will be used the same amino acids with already attached fluorescent dye?
Yeah, in fact, there are now already a number of techniques that were developed to do specific labeling of proteins. Okay, the example that I showed you, where we had this reversible dye binding to the membrane surface and then reacts by a non-bioorthogonal reaction, rather amide bond formation. So this example has only little specificity to plasma membrane proteins. However, it does not select specific protein or specific amino group or something. So now, but this method actually has a more specific examples, is when people use this one anchor group as a ligand that can recognize a receptor. So if I replace this guy with a ligand that binds, for instance, GPCR, then this group will react relatively specific to a completely natural amino acid, amino group. And this is called ligand-directed protein labeling. And so this gives you a very selective labeling. It's an emerging example, which is now used more and more worldwide, especially pharma people like it, because you can use ligand drug that can direct to the protein and then label it in the proximity, or called proximity labeling also. So now about artificial amino acid. Artificial amino acids, you can, in principle, introduce, for example, a click reactive group like azide or acetylene group. This requires a relatively tedious procedure when you wanted to put it in the cell. It is done on, you hijack actually their protein synthesis. So you synthesize a amino acid, a natural amino acid, and you put it in a transport RNA. And then this transport RNA will come and recognize specific codon and add it during protein synthesis. So you can do it also in vitro outside the cell. And so you synthesize big protein, which has a very specific azide group or acetylene. Now about fluorophore. If I put fluorophore instead of azide, the problem that the amino acid will be very big and much more different in terms of physical properties and chemical properties. And their enzyme may not recognize it anymore. You see? So if you make a very big amino acid, you won't be able to hijack biological process and because the enzyme will say, oh, this is not amino acid, I'm sorry, I cannot use you. And therefore, the modification should be minimal. And so this is people do the same with DNA, but in DNA, it is actually more efficient. So I saw examples where a DNA molecule was functionalized with quite a big dye, and then enzyme was still able to make DNA, and so you have a labeling. So it is possible to, but I would say orthogonal approach is more elegant and I think more efficient because the size is smaller. So amino acid is more like natural amino acid, you see. So this is minimal functionalization is very useful.
Thank you very much.
Yeah, you're welcome.
Yeah. Anatoly, by the way, the topic of research is very close to your topic, and it's a good opportunity.
Yeah, we even have one joint article in 2015.
Yes.
Yeah,
Yes,
but
exactly.
But you should continue your collaboration, I think. Okay.
Why not? I see there are a lot of interesting things, and Andriy do not need our help, but we are welcome if you.
But in fact, you saw there were many fellows, and some of them I didn't mention because I didn't talk, but about that works. But I collaborate and I'm open now, no problem. I would be happy to for student exchange as well and collaboration. I know there are very good schools on chemistry in Kiev and in Kharkiv, so and physical chemistry as well. So it's
yeah, you're right.
No problem. I'm open for collaborations.
Yeah, okay, Volodymyr Zaitsev.
Yes, thank you. Because everyone starts to talk about chemistry, I also have general question to you, Andrei. Do you see any difficulties now in Europe to find good organic chemists? Because you have a lot of students from Ukraine. In general, what do you think? Do we have more problem in the future with the students doing organic chemistry? This is the first part of my question. The second part, do you see any difficulties to do organic synthesis nowadays at the European University, saying about different limitations, waste treatment, and something like this?
Yeah. These are quite important points. So regarding students, I think for the moment there is no particular problem of finding organic chemists, especially if we have application, for example, in multidisciplinary field. So actually, their new generation students, they still like chemistry and organic chemistry, but many of them ask what would be the application. So, and that is something that fits to what I propose. And so usually we have students. However, it is the fact I said we have many Ukrainian students, but I have also many other nationalities. But it's true, I think Ukraine represents a majority. Then there are French students, Indian students, and other nationalities. However, I would say I have many Ukrainian students probably because of my origins and because of my connections with the university. And so people somehow know me, advise, probably they contact. But I also have to admit that Ukrainian students are really good. They have a good already experience. Some of them go through Yenamin, for example, and they already have a huge experience already after master diploma. They have more experience than many other students. And so in this respect, I wouldn't say, but it is true, it gets more and more difficult to get students in general. And good ones. Its selection is very difficult. But I don't think it's because of chemistry. Chemistry for them is fine because schools are still very strong. In France, schools of chemistry is very strong. They promote chemistry a lot, so it's all right. But the second question, that's indeed become more and more difficult. So chemistry is dangerous because toxic, because you have waste treatment, etc. And it is true the regulations in the Western world are getting more and more strict. And so at certain point, I would say, if we continue this way, maybe sooner or later, we won't be able to do organic chemistry because there are too many rules. And I give you a simple example is distillation. I remember when I worked in the department of organic and bioorganic chemistry in Kiev, distillation was the main method of purification. And we also needed to purify solvents for synthesis, dry them, etc. Here, it is forbidden to do distillation. Forbidden. We cannot do distillation, which is a fundamental way to treat the material. It is as simple as that. So now, to have a dry solvent, we cannot dry THF with sodium, etc. No, we need to buy a ready THF, which is not as good. And any kind of sort of distillations we cannot do. And regulations increase CMR products also, special places to do. So there are so many issues that increase over time, unfortunately. But I think people, after when they realize there are too many stupidities, they go back. And so it's fluctuating. So it's okay. I think chemists have some time still to exist in developed countries.
Is this restriction only in France or in other countries as well?
I think it is in all Western countries. They increased regulations, especially for materials which can be potentially cancerogenic. And also the waste treatment become very strict. So we have many type of wastes, which of course bases, acids. Then you have chlorinated solvents, non-chlorinated solvents, and then then biological waste is completely independent. And I think there will be many more type of waste which will. Of course, we have a special waste for chemicals, and they're also classified. And this is, of course, increasing the price of research.
Exactly, that you need to pay for any waste. And sometimes it's more expensive than to buy reagents.
Yeah, it's true. And so therefore, nowadays, we are no more looking at their price to quantity ratio, because before we were buying a bigger bottle because it is cheaper. But now we buy the smallest possible because utilization after to throw it is much easier. And so the logic is totally different now. Yeah.
Okay, very interesting information for me, because I visited a lot different universities in Germany, in Belgium, in France, and I made distillation that time and used other approaches, classical approaches, but okay, the situation changed.
Yeah, the situation changed, but this is quite recent. I think it's like 5 years ago they installed the rule. But we had some accidents, and then university decided it is on the local level. I don't think distillation is forbidden in Western civilization, no. but university was fed up of a little accidents, and so they say, okay, no distillation.
It was not the reason of consumption of water, of other. I believe it's because of the water.
But this is another story. Consumption of water is another issue. They actually nowadays they force us. They strongly advise us to use chillers, so which are circulating water, and it is better actually chiller, but it is expensive, of course, instrument. But in this case, you don't consume water at all, so you just circulate a liquid in the cooler. But that's really very practical. These cooling systems with some liquid. Yeah, but not because of water, no, because of the danger. In fact, if anything in the Western world is decided about chemistry, it's rather about danger, not about the not economic questions or ecological question. No, it is danger. This is the primary actually thing that drives the decisions.
Okay, thank you.
Okay,
Yeah.
thank you very much. Nice discussion. But I think at this point we can stop our recording.